Welcome to Lab, everyone. Today we're going to be talking about how we control microbial growth with chemicals. The last time we were together, we talked about one of the physical methods for controlling the growth of microbes, and that's ultraviolet radiation. Today we'll be turning our attention to the vast array of chemicals that are available to us both in professional situations and also in our own homes. Our objectives for this lab exercise are to first examine the four most common families of disinfectant chemicals. We're going to compare the relative effectiveness of these chemical disinfectants, and we'll really focus on some of the limitations that we find within these families of disinfectants. You're going to hear me say again and again during this laboratory that we have to know our chemicals. We have to know which chemical is the most appropriate to use in which situation, depending upon which types of microbes we're trying to control. Now, before we get into the specifics of classes of disinfectant chemicals, let's review a little bit of the vocabulary from the lecture about controlling microbial growth. Remember, there's a difference between the terms that we apply to chemicals that control the growth of microbes. There's a difference, for example, between a disinfectant and an antiseptic. A disinfectant chemical is going to eliminate common pathogens from inanimate objects and surfaces. So we are using disinfectants on things, not on our own bodies. If it's a chemical that we're applying to our bodies for the purposes of getting rid of pathogenic microbes, that chemical is called an antiseptic. So an antiseptic is going to also eliminate common pathogens, but we're applying it to living tissue, to skin primarily. Two other important terms to review here are bactericidal and bacteriostatic. We talk about these terms when we talk about antimicrobial drugs like antibiotics, but we can also apply them to chemicals. Bactericidal means kills bacteria. That's what the cidal part of that word means. That means death or kill. Bacteriostatic refers to stopping the growth of bacteria. So there are some chemicals that are going to be able to stop bacterial cells from dividing without actually without actually killing them outright. Remember, there's no single chemical that's going to be the best one to use in all situations. We have to match the chemical to the potential microbes and to the environmental conditions that are present in this place that we're trying to disinfect. We have to think about the surface that we're trying to disinfect. We have to think about things like pH and the solubility of the chemical. We have to think about how toxic this chemical is and who might be exposed to the chemical in this environment. We also have to think about the amount of organic matter that's present in the environment. And by organic matter, I'm talking about things like soil and grease. A chemical can be effective against one pathogen and be completely ineffective against another. You have to know your chemicals. One of the things that we have to do when a new chemical comes onto the market and wants to be labeled for disinfection purposes is it has to be evaluated. It has to be tested in professional laboratories to make sure that this chemical is going to meet the needs of the people who are applying it. If the manufacturer claims that it can kill certain pathogens, those claims have to be tested before that chemical can be labeled as a disinfectant. One of the ways that we can evaluate chemicals for their efficacy against certain microbes is by doing what's called decimal reduction time testing. We talk a little bit about this in the lecture. Decimal reduction time is shorthanded as DRT. In some source materials, you'll also see it listed as the D value for a chemical. What decimal reduction time is the time it takes in minutes to kill 90% of a sample microbial population. So however long it takes when you apply that chemical for 90% of the microbial cells to be killed, that's going to be your decimal reduction time. If you go into any type of source material that deals with disinfectant chemicals, that provides information about disinfectant chemicals, you're likely to run into tables that look like this. You can pull these tables up on the internet. These are important things to be able to find in terms of understanding the disinfectant chemicals that you're using. You'll notice on the left-hand side of the chart, the chemical itself is listed. You could see hydrogen peroxide at the top, 10% solution, betadine, 10%, chlorine, iodine, and so on. You'll also notice in the second column that there's a list of the particular organisms that were tested for each one of these chemicals. Pseudomonas aeruginosa, Bacillus subtilis, and Staphylococcus aureus are on this list. These three organisms were likely chosen because they have differing sensitivities to various disinfectant chemicals. You want to make sure that you test your disinfectant against organisms that come from a wide range of environments. You can see that they've listed the concentration that was tested. That is provided by the manufacturer. And if we move over among the last columns, here we see what's listed as D value. Remember, this is one of the ways that DRT is listed, D value. And again, the unit here is minutes. So let's think about what this means. If we're going to use a hydrogen peroxide solution of 10%, it's going to take 3.8 minutes of exposure time to kill 90% of Pseudomonas aeruginosa organisms. It's going to take 5.6 minutes to kill 90% of the Bacillus subtilis, and it's going to take 8.7 minutes of exposure to kill 90% of Staphylococcus aureus. I want you to think for a minute about how people tend to use disinfectants. Imagine yourself in your own home, and you have a bottle of some kind of spray cleaner in your hand, and you're going to clean and disinfect your countertop in your kitchen. Chances are, if you're like most people, you're going to spray that chemical onto the countertop and you're going to immediately wipe it off, correct? So you can see that it's really important in a professional situation, like a hospital setting or another healthcare type of setting. It's really important that the people who are doing the disinfecting know how to use these chemicals, because a lot of chemicals require an extended exposure time in order to be effective against a wide range of pathogens. There aren't very many places that would use hydrogen peroxide as a disinfectant. It's much more common as an antiseptic. But look at these exposure times. You would have to spray or place that hydrogen peroxide on top of a surface and leave it for 9 minutes if you suspected that Staphylococcus aureus might be a pathogen in that particular environment. That's a long time to leave a chemical sitting on a surface. So As I said earlier, and as I'll say again and again today, it's important that we understand our chemicals and we understand how they work so that we can use them effectively. In addition to the decimal reduction time, there are other tests that are performed on chemical disinfectants to make sure that they are effective for that particular use. One other one that I'd like us to be familiar with is called the use dilution test. And this is a test that's performed by a group called the American Official Analytical Chemists, or the AOAC. This particular test, this use dilution test, uses Salmonella and Enterica cholera suis as a test organism, as well as Staphylococcus aureus and Pseudomonas aeruginosa once again. Notice that this first organism has three names. Salmonella is the genus, Enterica is the species, and cholera suis is the strain. Don't forget that a lot of pathogens have strains associated with them as well. In the use dilution test, the operator takes steel cylinders, small steel cylinders called carriers, and I'll show you a picture of these in just a moment, and dips them into a broth culture. Those steel cylinders are then set down on some filter paper and allowed to dry. Essentially, why this is done is they are coating this steel cylinder in the organisms. They're coating the surfaces of the steel with bacterial cells. Those steel cylinders are then placed into disinfectant at the recommended dilution, again, according to the manufacturer, and for the recommended time, according to the manufacturer. The cylinders are then removed from the disinfectant and blotted and so on, and then placed into a nutrient broth, just a regular basal medium, in order to allow the growth of surviving organisms. And those surviving organisms can be quantified doing some math. Now, this particular test, this use dilution test, is only for bactericidal products because, of course, they are going to quantify how many cells have been killed by the disinfectant. The same would also be true for the decimal reduction time because that also measures death of cells. On this slide, you're looking at a picture of several glass petri dishes with filter paper placed in them. These are sitting underneath a biology or a biological hood so that we're in a sterile environment here. Notice these little steel cylinders. They're about an inch or so long. They are open. They have an opening through them. And these are what are placed down into the broth culture in order to coat them with organisms. There are several modified protocols that can be done for this use dilution test. For example, there is a modification where you can take an actual broth culture and expose it directly to a disinfectant. Some people prefer that version of this test because they feel that it allows all of the cells to be uniformly exposed to the disinfectant. The way that version of the test works is once the disinfectant is added into the culture, an aliquot or a sample of the culture is removed at certain intervals of time. And those cells are then placed onto an agar plate and put in the incubator, and the colony count is used to determine how many cells have been killed so far. All of these different versions have published protocols available for them. Some of them are purely qualitative and some of them are quantitative. It's important to note, though, that whether you're talking about the use dilution test or the decimal reduction time tests, These are tests that are only done when the manufacturer is claiming that the disinfectant is bactericidal. We're going to switch gears now and start talking about the classes of disinfectant chemicals that are available to us. There are 4 large classes that are most commonly used in healthcare settings and in other professional or commercial settings, as well as used in the home in order to control the growth of microbes. These 4 classes are the alcohols, the chlorinated compounds, the iodophores, and the quaternary ammonium compounds. We're going to talk about each family of chemicals separately. We're going to begin with the alcohols. There are lots of different alcohols available for disinfectant purposes on the market, including things like isopropanol or isopropyl alcohol, ethanol, and so on. Alcohols, if you recall from our early lectures in the course, alcohols are rapidly bactericidal, so they make great disinfectants for use in the microbiology lab. But you may also remember that they have to be diluted a little bit to be most effective. We use alcohols at a 60 to 90% solution. In most of the laboratories where I've worked, we use 70% alcohol as the primary disinfectant in the laboratory. Alcohol is rapidly bactericidal. That makes it effective even if it's just sprayed over the work surface and then immediately wiped off, which is how most people are going to use it. So it's a great choice as a disinfectant in a micro lab. It is interesting to note that it has to be diluted to be at its top effectiveness. A lot of people will think that if 70% alcohol is good, well, then 100% alcohol must be even better. But in actuality, it has to be diluted to be at its peak effectiveness. Undiluted alcohol is simply less effective. It turns out that the water that we add into the alcohol actually promotes the death of the microbial cells. Now, one of the things we'll talk about for each of these classes of disinfectant chemicals is what we call their scope of activity. And essentially what that is a measure of which particular types of microbes it's effective against. One of the most concerning pathogens in healthcare environments is the one that causes tuberculosis. As we've talked about, the Mycobacterium tuberculosis organism secretes a waxy substance called mycolic acid out around itself and makes itself more resistant to things like disinfection. So Mycobacterium is often one of the organisms that we are interested in terms of whether or not a chemical disinfectant is effective. When it comes to the alcohols, alcohols will kill this TB-causing organism. They will also kill fungi and viruses, as well as bacteria, as we've said. But it's interesting to note that alcohols are not effective against bacterial spores. Remember, bacterial spores are one of the most difficult things to destroy when it comes to controlling the growth of microbes. The mechanism of action of alcohols as disinfectant chemicals involves denaturing proteins. Remember, proteins are, at their most simple structure, a long chain of amino acids that are bound together with peptide bonds, which are a special type of covalent bond. Once that long chain of amino acids is assembled inside of a cell, it then has to fold. and it folds up using weak attractions like hydrogen bonds and van der Waals interactions. That creates a three-dimensional functional protein. When a protein is denatured, it's those weak attractions that are broken down. So the chain of amino acids is still intact, but it's no longer folded up. And if it doesn't have its three-dimensional shape, it can't do its job. What alcohol does is denature. It unfolds proteins and makes them ineffective. That's going to lead to the rapid death of the cell. Now, there are some limitations to the use of alcohol, some things that are important for us to know. Alcohol, like a lot of disinfectant chemicals, can damage certain materials. In particular, it will damage rubber over time, it will damage certain kinds of plastic over time, and it will damage lenses over time. So it's important to know, for example, that you don't want to put your alcohol disinfectant onto your objective lenses on your microscope. You want to use specific compounds that are designed to clean objective lenses. Another interesting thing to note about alcohol is that it's expensive. A gallon of concentrated alcohol, like this one in the picture, costs about $100 right now. Now you are going to dilute this down to about, again, 70% or so, but that's an expense that some facilities don't want to take on, particularly if they're disinfecting a large area. So while we often see alcohol in laboratory spaces as the disinfectant, that doesn't mean that that's the only chemical that's out there and being used. On this slide, you're simply looking at an artist's depiction of this process that we call denaturation. On the left-hand side, you can see a folded protein. Remember, all a protein is at its most basic is a long string of amino acids, and that's what this green ribbon represents. That long green ribbon is going to start to fold up. Different R groups within each of the amino acids are going to be either polar or non-polar. Some of them are acidic, some of them are basic, and they're going to be attracted or repulsed from one another, creating these shapes and structures that you can see here. This represents a three-dimensional molecule. And that three-dimensional molecule has one specific job to do. Its structure is related to its function. If you denature that protein and you break down all those folds so that now you're left with this chain of amino acids, this protein cannot do its job. So alcohols are really good as disinfectants. They are rapidly bactericidal. They're effective against Mycobacterium tuberculosis. They are effective against viruses and fungi, but they will not destroy this thing, this bacterial spore. Remember, the spore protects the DNA in the core. The vegetative cell of a bacterium sporulates, if it's able to, in order to save itself from sure destruction. The DNA from the cell is preserved in the core, and the rest of the cell goes away. The DNA that is then surrounded by all these different layers of material that protect it from almost everything. Spores persist in the environment for as long as we can imagine. Remember, we have collected spores from the wrappings around mummies that were buried thousands of years ago, and those spores have been able to revert back to the vegetative state when they were brought into the laboratory. Alcohol does not destroy bacterial spores. The second family of disinfectant chemicals are the chlorinated compounds. The best example of a chlorinated compound in all of our lives is bleach, plain old household bleach, or what is technically called hypochlorite. I have a picture of just a generic bottle of bleach down here. We can use bleach in our laboratories and professional spaces as a disinfectant. And of course, people also use bleach in their homes. Like alcohols, chlorinated compounds are rapidly bactericidal. So they can be sprayed on a surface and wiped off almost immediately and still be bactericidal. We dilute these compounds to a 10% solution, remember. That's when they will be most effective. In terms of the scope of activity, they are effective against the organism that causes TB, yes, but you have to put the bleach down onto the surface and leave it for several minutes. So that is a limitation for the use of bleach, at least in settings where you are suspicious that you're dealing with contamination with mycobacterium. Chlorinated compounds are effective against fungal cells. They are effective against viral particles, but again, they require minutes of exposure time. And finally, chlorinated compounds will destroy bacterial spores, but you have to put down a higher concentration, higher than 10%, and you have to leave it for minutes of exposure time. One of the most interesting things about these chlorinated compounds is that the mechanism of action, how it is that they are killing cells, is not completely understood. We do know that when you put something like bleach into water, when you dilute it with water, it becomes an oxidizer. And what I mean by that is it's an acid that's able to steal electrons from other molecules. Remember we talked about radicals? Well, this particular compound becomes an acid when it's placed into water, and that particular acid is an oxidizer. It can take electrons away from other molecules and thus damage those molecules and kill the cell. Now, there are limitations to chlorinated compounds, just like there are limitations to all disinfectant chemicals. First, chlorinated compounds are less effective when they're used in an environment that has a high pH. So if the environment that you're trying to disinfect, the surface you're trying to disinfect, has a higher pH associated with it, that's going to be problematic. And that makes sense if you think about it. If the effectiveness, or at least part of the effectiveness, of something like bleach is that it becomes an acid once it's in water, if you were to then expose that acid to an alkaline environment, that then is going to neutralize the chemical. Acid plus alkaline becomes neutral. Bleach is also famously less effective in the presence of organic matter. So remember, by organic matter, we mean soil, dirt, grease, anything like that. So if you're going to use something like bleach as a disinfectant chemical, you have to clean the surface first. This is particularly important in the home, if you think about it. If you're going to use bleach as a disinfectant in your kitchen, it's especially important that you clean off the surface first and then disinfect it with the bleach. There are lots and lots and lots of compounds in grocery stores and other retailers that are sold to homeowners as disinfectants and that are based on bleach. The Clorox company alone has probably a dozen different chemicals for sale that are labeled as disinfectants to be used in the home. So it's important that people understand that these compounds are going to be a lot less effective if they are sprayed directly onto dirty surfaces. Bleach is also known for damaging materials. You probably are already aware of the fact that bleach will discolor fabric if it's sprayed onto fabric. Now, obviously, we take advantage of this in the laundry. Bleach is often added to white materials in order to brighten them because it does take colors, dyes out of fabrics. The other thing that bleach will do, though, is corrode. Bleach will eat away at metal surfaces over time. So if you're in a facility or a professional environment or a laboratory where you do have metal surfaces that are being routinely disinfected with bleach, they're going to pit. They're going to corrode and break down over time. One of the things that has been in the news very recently, if you've been listening to the news, involves, unfortunately, this particular limitation of bleach, of chlorine, I should say. I'm sure you've all heard the news about the terrible collapse of the Surfside condos down in Florida. A partial collapse of this building took place several days ago now. Um, there's going to be a thorough investigation as to how this building just collapsed in the middle of the night. But one of the pieces of evidence that has come forward is that there were some structural deficiencies in this building. Even though the building wasn't all that old, the building was first erected in the 1980s. Like a lot of places down in Florida, a lot of condos, this one was set right on the edge of the ocean and was subjected to the spray from the ocean and the high concentration of salt that is in seawater. This building was primarily made from concrete and rebar, like a lot of condos are down in Florida. So some people are suspicious that exposure to seawater over time may have corroded again the concrete and the rebar inside the concrete, the steel inside the concrete. Well, something else that has come to light is that the pool that was present down on the ground level of this condo, the pool had inadequate drainage. And of course, pool water is disinfected with chlorine. And there are images that are coming out now on the internet that show that pool water that, would be splashed out of the pool during routine usage of the pool was in fact flowing over into the parking garage area, which is where the collapse first occurred. At least that's the opinion of some of the experts so far. It is possible, although it has not been proven yet, it is possible that the buildup of pool water in the underground parking garage and the chlorine within the pool water weakened the concrete because chlorine will corrode over time. So I'm fearful that there's going to be a long list of structural deficiencies that are going to come to light in the investigation into why this building collapsed. But it is interesting to know that there's at least some suspicion that inadequate drainage of that chlorine containing pool water may have played a role. The last thing I want to mention about the use of chlorinated compounds as disinfectant chemicals is this one. It's very important that people who handle chlorine understand that you cannot mix chlorine with ammonia. Ammonia is not nearly as common anymore as it used to be as a household disinfectant, but people like my mother and my grandmother, for example, would have used a lot of ammonia in their home as a disinfectant. When you mix ammonia-containing compounds with chlorine-containing compounds, you get a toxic gas liberated That gas is called chloramine, and that gas will kill you within minutes. Every year, there's at least one case in the news of someone mixing disinfectant chemicals and dying from exposure to chloramine. There was a case, I want to say 2 years ago now, of a worker inside one of these chain restaurants like Chili's or Applebee's. And this poor person was cleaning the restaurant after hours and mixed these two chemicals together, creating this toxic gas. The person then collapsed. The manager of the restaurant came in and found the person and then collapsed, and both of those people died. So it's really important that we get that word out that you don't want to mix disinfectant chemicals because you don't know how they're going to react with each other. And the mixing of chlorine and ammonia is particularly toxic. The last thing I'll say about chlorinated compounds is that they're a lot cheaper than alcohol. If you go to buy a gallon of household bleach in the grocery store, it's a couple of dollars. It's not that expensive. Compare that to the $100 per gallon cost of alcohol, and you can see why bleach is often used in facilities where they're going through a large volume of disinfectant. The third family of chemical disinfectants is the iodophore family. Now notice this term is not iodine, it's iodophore. And what an iodophore is an iodine compound that has been mixed with what's called a solubilizing agent, something that's going to help the iodine be more effective. Remember when we talked about gram staining and Graham's iodine, we said that iodine is helpful as a mordant because it's very good at penetrating material. The same is true for these iodophore compounds. Now it's important to note that iodophores are used much more commonly as antiseptics than as disinfectants. So it's very common to see iodophores in the healthcare setting, but not as disinfectant chemicals, rather as antiseptic chemicals, something that would be applied to the patient's skin for the purposes of killing microbes. One of the most common iodophores on the market is one that goes by the brand name betadine. There are also lots of generic types of betadine out there, and those are simply referred to as povidone iodine. So if you see a container that's labeled povidone iodine, that is a generic brand of betadine. Now, iodophores are bactericidal, which is why they make good antiseptics, but they're not as rapidly bactericidal as alcohol is. Alcohol, as you know, can be used as either a disinfectant or an antiseptic. We use alcohol a lot as an antiseptic in healthcare settings. Now, when it comes to the scope of activity, iodophores are effective against Mycobacterium. They're also effective against fungal cells, but you have to leave the iodophore on for a couple of minutes. Iodophores are effective against viruses, and they are effective against spores, bacterial spores. But again, it requires minutes of exposure. The mechanism of action of iodophores involves denaturing proteins again and damaging the DNA molecule. So like alcohols, iodophores are going to denature or unfold proteins. But they have the added mechanism of also damaging DNA, and that's because they're able to penetrate deep into the cells of these microbes. Now, iodophores have some limitations, as all disinfectants do. Believe it or not, there are some species of bacteria that can survive inside a bottle of betadine or povidone iodine. The take-home lesson from that is that if you're going to mix up and use iodophores in your facility or your laboratory, it's important to change your solution frequently. I forgot to mention it earlier, but you're going to also want to do that for your chlorinated compound solutions. When you make up a disinfectant solution of bleach, it becomes less effective over time. The general recommendation is that you have to make up a fresh bottle of the diluted bleach once a week because these compounds degrade over time. So both bleach and iodophore solutions have to be made up fresh periodically. On this slide, you're looking at a drawing of a membrane of a bacterial cell. You can see the heads and the tails of the lipid bilayer. You can also see proteins drawn inside the membrane. This area represents the inside of the cell. You can see the DNA molecule shown here and some other proteins. This area out here represents the outside of the cell. And what the artist is showing us is that the povidone iodine comes in looking something like this. You can see that the iodine itself is within this solubilizing agent. The iodine molecule will then be liberated from the solubilizing agent, and it's able to penetrate through this membrane. So it's going to affect both proteins and the DNA molecule inside of microbial cells. The last group of chemical disinfectants is the quaternary ammonium compounds. These are compounds that are composed of an ammonium ion and what's called a modifying agent. Again, these agents that get added in to these chemical solutions are designed to make whatever the active ingredient is more effective. So we add a solubilizing agent in with iodine, and we add what's called a modifying agent in with the ammonium ion. There are lots of quaternary ammonium compounds on the market, both disinfectant chemicals and antiseptic chemicals. This is an antiseptic that you've probably seen if you've worked in healthcare at all. That's Hibiclens. Hibiclens is one of the most popular quaternary ammonium compounds that's sold as an antiseptic. There's another very common brand called Rocal. There's one called Virex, which is used in hospitals and long-term care facilities, again, as a disinfectant. There's also a generic type of quaternary ammonium that goes by the name chlorhexidine. That's actually what Hibiclens contains as well. When it comes to the quaternary ammonium compounds, they are bactericidal, but not nearly as rapid as alcohol and bleach. In terms of the scope of activity, they are not effective against the organism that causes tuberculosis. That's important to know. They are effective against fungal cells. They are effective against viral particles, but only enveloped viruses. Now, if you remember from the virology lecture, when it comes to naked viruses, we're primarily talking about plant viruses. When it comes to animal viruses, or viruses that infect animal cells, they're usually enveloped. So this is not really a limitation, since we're using the quaternary ammoniums to protect humans against these microbes. So it will be effective against viruses that can infect human cells. quaternary ammoniums are not effective against bacterial spores. And when it comes to the mechanism of action, they are doing two things to these cells. They are denaturing protein, you see that one again, and they're disrupting membranes. Quaternary ammoniums are able to break apart cell membranes. That's why they're particularly effective against enveloped viral particles because, of course, the envelope is made out of membrane. On this slide, you're looking at an artist's depiction of a microbial cell. And what the artist has done is taken a little piece of the membrane and blown it up for us in this image. These little red circles with the two black sticks coming out of them, these represent ammonium ions, and these have a positive charge associated with them. When they interact with the plasma membrane, they break it apart. And that's what the artist is showing us here. So these are compounds that can unfold or denature proteins, as well as breaking or disrupting cell membranes. When it comes to the limitations of the quaternary ammoniums, there's a couple of things to note. Probably most important is that they are less effective in hard water. There are different parts of the country where hard water is a problem. There are some parts of the country where the water is particularly soft, and there are other parts where the water is particularly hard. And generally speaking, when we talk about the hardness or the softness of water, we're talking about the amount of minerals that are in the water. So when you try to mix up these quaternary ammoniums in hard water, the minerals in the water bind up the ammonium ion, and therefore the compound becomes less effective. Interestingly, quaternary ammoniums are less effective as antiseptics if you apply them with cotton. I find that fascinating. So if you were going to use, say, Hibiclens as an antiseptic, let's say prior to a surgical procedure, you would want to make sure that you were applying the Hibiclens with a sponge that was not made out of cotton, because cotton actually binds up the ammonium ion and makes the Hibiclens less effective. Very interesting. Here's another really important thing to note about quaternary ammoniums. Now, this is more related to their use as a disinfectant, because when you buy a product like Rocal or Virex, which is what I'm showing here in the picture, these are concentrated forms of the quaternary ammonium, and they have to be diluted before they're used. People who do this work, so the janitorial staff, for example, in a long-term care facility or in a hospital setting, the people who are exposed to the concentrated forms of quaternary ammonia compounds, they can become sensitized over time to these products. And by sensitized, I mean they develop respiratory symptoms after being exposed to these concentrated, again, quaternary ammonium compounds. So the person who is exposed to these compounds, to the vapors of these compounds, again, over time, can sometimes start to develop a respiratory reaction to those vapors. And that's what you're seeing in this drawing here at the bottom of the slide. You're seeing an image of the human trachea and the bronchi that branch off of the trachea, and then the human lung. And if you'll notice on the left-hand side, you've got this bronchus that looks perfectly normal. The lining of the bronchus is narrow or thin, and there's a large airway inside the bronchus. Now compare that to this side, where the lining of the bronchus has become very inflamed and has started to secrete this material. This is what we refer to as a sterile inflammatory response. This is a response to the chemical itself. those vapors cause this sensitization. It's a immune reaction inside the airways. And as you can imagine, this is very problematic, especially for someone who might already have a respiratory problem like asthma or COPD. If their airways start to swell when they get exposed to the quaternary ammonium, they could be in trouble. So something important to note. All right. So we're not together today for me to take questions from you, but if you have any questions or comments about anything that we've talked about in today's laboratory exercise, you can message me directly, or you can post it on our discussion board called Questions, Questions, Questions. And I will see you again in our next laboratory meeting.